So, good morning, everybody. Today, I will be talking about the pituitary gland in fishes. As such,
Good morning, sir.
good morning, good morning. So, as such, there is not much difference between the pituitary glands of a human being and the pituitary gland of a fish, but there are certain minor differences. And of course, you need this sort of information is very important for your net studies as well as your other studies as well. So, in this talk today, uh, I will be talking from one of the books which I will be very shortly I'll share in the Google Classroom, or possibly I have shared this particular chapter already in the Google Classroom. Have a look. So I think that is good enough to study. Enough information is there in from S.S. Khanna. That's one of the best fish books I've ever seen. That is good enough for your studies and for competitive exams also regarding fisheries. So, I will start sharing the screen. Somebody is joining, it seems. Let's make that person enter. Okay. So, this falls under this category, endocrine and neurosecretory system. So, the first thing which we will be talking about is on the pituitary gland. Somebody is joining. Pituitary gland, as you know, is also known as the hypophysis. It is the most important endocrine gland in any organism, not only in teleos but in any organism. It is also known as the master gland. It is known as the master gland. And as you know, this is also divided into new the neurohypophysis or posterior pituitary and the anterior hypophysis or adenohypophysis. The neurohypophysis is mainly made up of nerve cells, nerve actually nerve cell nerve endings basically, and this region is glandular. The real endocrine part. Of course, even the neurohypophysis also has got vasopressin, oxytocin, as we know, it's been secreted over here. Okay. As we know the thing that pituitary is situated just ventral to the brain behind the optic chiasma. You have read if you look at the brain of a teleost, just the cross section in the mesencephalon, you can find an optic chiasma over here. And this optic chiasma, below this optic chiasma is the, you have your pituitary located. Optic chiasma, where the two optic nerves they cross each other. They cross each other. The optic nerves they cross each other. Now this is the structure of the pituitary gland. What you find over here is that this region, this is the neurohypophysis, and this region is the adenohypophysis. And interesting among this, the shape is a bit different from our mammalian pituitary. There are different types of cells which are present, you must learn. The corticotrophs, the corticotrophin hormone, adrenocorticotropic hormones, then the thyrotrophs, these are the thyrotroph cells. Then you have these prolactin cells which are labeled. Then you have somatotroph cells which are there. And you can divide the adenohypophysis into the regions known as the pars intermedia, this region, pars distalis, this region, and the distal or rostral pars distalis. This is the proximal pars distalis, and this region is the rostral pars distalis. So this is essentially how the pituitary gland looks like. And you know, of course, one thing that this is the stalk or infundibulum. It is absent in some species. It is absent in some species. Now what you have seen where the rostral pars distalis is. So this is the region of the rostral pars distalis. This region. So now let us talk about what's there in the rostral pars distalis. So this is the anterior most part of the adenohypophysis, and three types of cells are present. Most of the cells are acidophils and are stained by azocarmine or orange G. These cells may show follicular arrangement surrounding the blood. Follicle means goal structure, round structures. These are follicular arrangements. A few cells are stained with aniline blue and aldehyde fuchsin, past stainings, and are called cyanophils. A few non-granular cells that do not stay are known as chromophobes. They don't take any color. Some take color, acidophils, acidic stain. Some take aniline blue, which is basic, and they of course take aldehyde fuchsin, and they are known as cyanophils. And some are, they don't take color, they are chromophobes. Then you have the proximal pars distalis. This is between the proximal rostral pars distalis and the pars intermedia. I had shown the picture there. It's in the intermediate region, but is not clearly demarcated from the former. It can be differentiated from the pars intermedia due to large ramification of pars nervosa, that is from the lot of nerves from the neurohypophysis, they innervate the pars intermedia, but in the distalis proximal, proximal, it is not so much innervated by nerves. The PPD or proximal pars distalis, it contains acidophil cells, cyanophils and chromophils, and they may vary in size during different parts of the year. During maturation and spawning period, the reproductive times, you find a lot of cyanophil cells present there, and rest of the year the acidophils are larger in number. There are more acidophils in rest of the year. During maturation and spawning periods only you have lots of cyanophil cells. They can be stained by azocarmine or orangey stains, the acidophils, and the cyanophils, they are stained by aniline blue, PAS, aldehyde fuchsin stain, and they can be also divided into further categories on the basis of size, shape and staining affinities. And as usual, the chromophobes do not take any stain. So there is one very minor difference: during maturation and spawning period, the cyanophil numbers, they increase a lot. Let's go to the next region, which is the pars intermedia, absolutely the one on the left. Let's go to the picture and I show you once more. So the pars, this region, this is the pars intermedia. This region is the proximal pars distalis, and this region is the rostral pars distalis, and the neurohypophysis is this region. And as you can see from this picture, in the pars intermedia, there is lots of nervous ramifications. These are all nerve ramifications. Then we go to the pars intermedia. It is the posterior part of the hypophysis, and it is extensively supplied by branches of the pars nervosa. There may be various sizes in different species, and they consist of two cells: cyanophils that are stained with aniline blue, and acidophils that are stained with azocarmine or orangey. Some species, also called amyphil cells, that take both blue and orange stain. So acidic and basic stain, they take both of them. And there are some chromophobes which don't take any stain at all. The cyanophils, as we know, are negative to PAS, AF and aldehyde, and when the pituitary is stained with PAS followed by lead hematoxylin, PBH, two distinct cell types are observed in some species. You must know what are the different stains being used. This is the only difference which you knew. Since you had histochemistry, you have to know the different types of stains. Of course, you know the different type of cells, but you need to know the different types of stains. In your second semester BSc, you had pituitary, you had read about some of these chromophils, acidophils, but here you have to know about the stains in much details. There you had to expect only 1 or 2 marks. Here you may expect a higher amount of marks. So PAS positive cells are oval in shape and are situated near the border of the neurohypophysis, while the second type of cells are stained with lead hematoxylin. The pars intermedia is well vascularized and is believed to secrete distinct hormones. Two distinct hormones. They secrete two different hormones. So this part deals with your pars, the adenohypophysis part. Now let us go to the neurohypophysis part. In the neurohypophysis part, a large number of nerve fibers arise from cells in the hypothalamus. So basically, the endings of the below the hypothalamus is the hypophysis or the pituitary. It forms an axis known as the hypothalamo-hypophyseal axis. It is called. So the hypothalamus has got neurosecretory centers which whose endings end in the neurohypophysis, and these are neurosecretory nerves. These fibers branch repeatedly and are intimately connected with the pars intermedia. Like I told that from the neurohypophysis, this is the, say for example, the pars intermedia of the adenohypophysis, and this is the neurohypophyseal region. You have extensive nerves, and they all come from the hypothalamus region. Hypothalamus. Hypothalamus region. And this is also known as the hypothalamus hypophyseal system. Or if you talk about the thyroid, you can call it as some people call it as the HPT axis, the hypothalamus pituitary thyroid axis, or HPA axis, hypothalamic pituitary adrenal axis. You must have read all these things in physiology. So basically, they are essentially the same. What you see in mammals and here it's the same. So pituicytes and droplets of neurosecretory materials are seen in these fibers. You find lymphocytes and migrating cyanophils. They can be stained by aldehyde fuchsin, alum hematoxylin, and aldehyde thionin. And there are some bodies, large droplets of neurosecretory materials are seen, and they are known as herring bodies. They are stainable and non-stainable fibers have been distinguished depending upon their stainability with aldehyde fuchsin and other stain. So the important thing is neurosecretory materials are synthesized in the hypothalamus and transmitted to the pituitary through fibers. So this is the mid-sagittal section, the mid section. What do you find over here? You find this is the pars intermedia region, and you know this is this part is the rostral proximal pars distalis, and this is the rostral part distalis. This is the infundibulum or stalk which is coming out, and there are lots of nerve fibers which innervate the pars intermedia. This is what I had been explaining so long. Now comes the interesting, what are the cell types and hormones of the pituitary gland. So let us talk about the gonadotropes. If you see the picture, you will find pictures, cells known as gonadotropes, somatotropes. So what are the function of the gonadotropes? They are also known as the FSH and LH cells. These cells are mainly situated in the proximal pars distalis. But in some cell species like eel and trout, they migrate to rostral pars distalis at maturity. So again, as usual, you have to read about the stains. They contain glycoproteins and are stained with PAS, Alcian blue, aniline, and AF. So the question may come: what are the different stains by which you can stain glycoproteins in say in the pars proximal pars distalis? This question comes. So you may be given an option of PAS, Alcian. You must write PAS, Alcian blue. So maybe 2 marks. So write 2 stains by which you can stain glycoproteins. So you should be knowing PAS, Alcian blue, aniline blue. You can give 2 examples. So these type of questions are very much expected. You may find it. And let me tell you that it is not me who sets question papers. It may be anybody in the university who can set this question paper. If it's me, then it's me. I am me from the BB college. We are just simple teachers. Okay. So the two hormones are secreted. You know, the follicle stimulating hormone and the LH hormone. And so there are some histocytological changes in correlation with the maturation of gonads. So I had explained this before. So these are the types. You know the function of FSH and LH. I think this is your homework. You must find out what is the function of FSH, follicle stimulating hormone. They help in maturing the egg, and luteinizing hormone, they help in rupturing the egg from the follicle. From the follicle, the egg comes out because of LH stimulation, luteinizing hormone. And follicle stimulating hormone, they mature the follicles and the egg is inside the follicle. Then the empty follicle then becomes into the corpus luteum and secretes progesterone, which maintains the pregnancy like state. So you people must be knowing about this very well. It's the same, the physiology is just the same, in whether it's a fish or whether it's a rabbit or whether it's a human being. They are 90%, they are the same. So if you give the function, if you don't know the function in a fish, you give the function of in a mammal, then of course you'll be at least 90% right, and in our current examination system you'll be given full marks. So basically, therefore they are named the same. Question, homework for you: what is the function of FSH and LH in males? This is a thing which you need to find out. What is the function of FSH and LH in males? This is something which you must do a literature work and find out. So the next type of cells which we go across are the thyrotrop cells or the TSH cells. They are found in the proximal pars distalis and they are basophils and cannot be easily distinguished from the gonadotrops on staining affinity. So you must study about TH. They are aldehyde fuchsin positive. Gonadotrops are aldehyde fuchsin negative. So by staining, how can you distinguish between THS cells? Okay, you say that THS and gonadotrop cells. TSH cells are aldehyde fuchsin positive, while gonadotrops are aldehyde fuchsin negative. So these types of questions also you can expect. So the next cells which we go are the somatotrophs or STH cells, growth hormones. They are important for the growth. Uh, mm, you can increase the length of the teleus by growth hormone by increasing appetite and protein synthesis and by stimulating this release of insulin. And you must also read about all the stains which are present here. The orangey stains are somatotrophs. Cells taking orangey stain are somatotrophs. They are found in the proximal pars distalis. The second type of cells, which are the acidophils, which take azocarmine or erythrocin, and they are the prolactin cells. So difference between prolactin and thyrotrophs on the basis of the staining, the answer is located here. The somatotrophs also secrete growth hormones which influence growth of various tissues. So these are essentially of what you see of the somatotrophs. Then you have the corticotrophs, the ACTH, adrenocorticotropic hormone. You know this hormone very well. So they are situated in the rostral pars distalis and the neurohypophysis, and are believed to secrete these ACTH cells, adrenocorticotropic hormone. They are elongated and chromophobic without granules, but they can be stained faintly red with azocarmine or erythrocin in some species. So the staining part is something which you must remember. This is the something additional which you must study from your BSc knowledge. Then the prolactin cells, they are also known as lactotrophs. The question, what are lactotrophs? So I've written it slightly what are what cells, just in case, my little notes in from my book. The book is actually not with me, it is with one of my students who has not yet returned, but she will return because the lockdown came and we could not meet and she was unable to return it. But I ask her and then she takes a photo and sends it to me. And she is one of my favorite students also. So, um, then you have the prolactin cells. They can be sustained with erythrocin, azocarmine, or acid fuchsin, but they are negative to PAS, Alcian blue, or AF, aldehyde fuchsin. So you have an acid fuchsin and an aldehyde fuchsin also. There are two types of these fuchsin stains. Please read the principle of PAS staining. Pankaj sir, I guess, has gone through these very well. So prolactin, which initiates the secretion of milk in mammals and secretion of crop milk in birds. But here's the question: what is the function of prolactin in fishes? মাছের দুধ হয়? Do fishes secrete milk for their young? Obviously they don't. They are not mammals. So in fish, prolactin is involved in osmoregulation and involves the movement of water through sodium through gills, kidneys, and urinary bladder. This is a very important question. What is the function of prolactin in fishes? This question, I bet, this is a very favorite question which comes. Two favorite questions which I see of people asking is, what is the function of FSH and LH in males, and what is the function of prolactin in fishes and birds? I remember one student writing, I asked what is the role of juvenile hormone. So, juvenile hormone helps in menstruation, in insects, somebody has written. So, these things, kindly don't make these mistakes again. These type of mixture. It's not yours, the batch after you. But that student was a good student, and that's the reason why it's very surprising that even good people make so much silly mistakes. Okay, there are two hormones of the pars intermedia and neurohypophysis, MSH and MCH, melanophore dispersing hormone and melanophore concentrating hormones. Then of course, the vasopressin and mesotocin. This is actually not exactly like oxytocin, similar to oxytocin, but it is mesotocin. And what is the in mammals, oxytocin stimulates uterine muscles and increases secretion of milk from mammary glands, while the vasopressin constricts blood vessels and enhances water retention by kidneys. In teleos, the hormones of the neurohypophysis are believed to control osmoregulation by influencing gill permeability. This is an important place for questions. You know that in mammals, but in fishes this is different. So therefore I am highlighting this region much more. The next part comes is the neuroendocrine regulation of the pituitary gland. So as we know, it's a master regulator, and so it controls a number of metabolic activities of the fish, but the secretion of hormones regulated by the hypothalamus of the brain. The nucleus preopticus, NPO, and the NLT, lateralis tuberis, are important nuclear areas which control the activities of the pituitary. It is located dorsal to the optic chiasma. Ventral to the optic chiasma is the pituitary, and dorsal to it is the NPO of the hypothalamus. Then you have to the NLT lies along the ventral surface of the infundibulum or the stalk, and its neurons innervate the cells of the hypophysis. The hypothalamus controls the activities of the pituitary gland either through these axons directly or transferring neuroendocrine hormones through a hypothalamo-hypophyseal blood portal system. So, what does this mean? It means directly the nerves go and they stimulate the cells, or from the hypothalamus there is a secretion which is secreted in the blood and some type of hormones, releasing factors, releasing hormones, they are secreted, they act on this, and they stimulate these the hypothalamus cells. You have known that, say for example, this corticotropic releasing hormone CRH, that stimulates the ACTH in the cells in the adenohypophysis, that therefore that secretes ACTH. ACTH goes through the blood and it stimulates the adrenal glands. The adrenal glands, of course, you know what they do. Adrenaline, noradrenaline, that is the medullary hormones, but the cortical hormones, the cortisone, corticosterone, all the cortisol, all these steroids are released from the cortex, adrenal cortex. So these things you have read already. As you know, as I was speaking, that the gonadotropic releasing hormone GnRH have they are so hormones of the pituitary gland, and they are regulated by the GnRH. GnRH is different regions of the teleostean brain, especially in the hypothalamus region. Besides a gonadotropic release inhibiting hormone GIH is also present in the brain of teleost. Then you have the thyroid stimulating hormone TSH of the pituitary. It's also controlled by a thyrotropin inhibiting hormone of the. So then you also it's the same ACTH, TSH, CR releasing factors. If you read the your semester 2 UG, you will find that you have the hypothalamus which secretes certain releasing hormones, RH, releasing hormones, GnRH for example. Then it acts on the pituitary adenohypophysis, on the special cells, ACTH secreting cells or TSH secreting cells, secreting TSH and ACTH. They eventually go to the thyroid or adrenal respectively, and they do their function. You can see the effects. So this is what's basically written over here. And you have the function of prolactin, which is different. The function of prolactin is different here in fishes. This is where the difference is. Extracts of pituitary gland, you know, they are used extensively in induced breeding of fishes in cultures. So first thing you must visit your second semester notes, function of the important pituitary hormones, and what are the difference. Just you could write it down. Go to your second semester notes, functions and the different types of hormones in mammals from the pituitary and hypothalamus. To that you must study the different types of stains in fishes, different types of cells and the different types of staining methods, and the stains which color different types of cells in the pituitary, as well as you must know the differences between some hormones like prolactin, FSH, LH, what is the function in fishes and in mammals. You must know the difference between them. So these are the things which you need to study in this essentially in this part. So this is I stop my sharing over here. I hope it's clear to people. Do you people have any questions? কোনো প্রশ্ন আছে? শেষ করার আগে জিজ্ঞেস করছি, if you people have got any questions. No questions, then I end my class over here.